How do enzymes function? Here's a schematic showing an enzyme with some distribution of charges on the inside. Could be +1, -1, or more likely these are just weak charges that are associated with hydrogen bonding. Here's a substrate that would fit, in contrast to a substrate that would not fit. The charges don't match. Or a substrate where the charges do match, but the shape is wrong. So this substrate can fit, the charges match up. If random molecular motion is moving this enzyme back and forth, remember collisions with water molecules and other molecules are constantly making proteins flex. When this stretches, charges here and these charges here are tending to pull the molecule apart. So if the molecule flexes enough, the enzyme flexes enough, then that can cause a covalent bond here to break. Now we have two products that can leave. For enzymes that function at normal body temperature in an animal, these reactions can go in both directions. You can have the two products be two substrates that bind, and the normal flexing of the enzyme when it's in this open state inward can push these two together, ram them together so that they fit tightly enough that they'll form a new covalent bond, and then you have formed the product of the reaction going in the other direction. In my classes, one of the examples of these would be an enzyme that converts H2O and carbon dioxide, water and carbon dioxide, into carbonic acid, a weak acid. The enzyme is carbonic anhydrase. It's an important enzyme involved in adjusting the carbon dioxide in your body. Carbonic anhydrase can take carbon dioxide and turn it into carbonic acid, or it can take carbonic acid and turn it into water and carbon dioxide. Charge does not have to be involved. We could have an enzyme that has just shape for molecules that don't have any charged residues on them, in which it's entirely shape that causes them to fit in. Again, flexing back and forth can cause bonds to form. In this case, flexing in this direction could push these two close to each other, such that we formed a new covalent bond and wound up with a molecule that looked like this, with the new covalent bond right here. What about enzymes that require an ATP to function? So some substrates may fit in a binding pocket, and I'll use some different charges here. They may fit in a binding pocket, but the normal flexing of the enzyme is not enough to tear them apart. Those are reactions that require a phosphate. An ATP is bound briefly. ADP, adenosine diphosphate, is left, but leaving behind a phosphate group, PO4 - 2. The power of this bond is actually the power of this phosphate group. If we put a -2 charge over here, and somewhere on another part of the enzyme is a -2 charge from two negatively charged amino acids, these charges repel each other. So this part of the enzyme being repelled from this negative charge and this part of the enzyme put tremendous additional force on our substrate here. And that then can tear the substrate apart as this enzyme flexes so that you get two products. Similarly, you can have an enzyme that maybe has binding pockets like this that binds two substrates, and the normal flexing of the enzyme is never enough to move those two together. But if there's a couple of positive charges over here, and we put a phosphate group -2 charge over here, that -2 charge is going to be strongly attracted to this positive charge, which can pull this whole thing around so that these two substrate molecules get combined into a single product. So the power of a phosphate bond is in causing a dramatic and sudden and powerful rearrangement of a protein because of repulsion of negative charge or attraction to positive charge.